So what Mendel did, he took pea plants. He actually looked not just the color of the pea plants, but he also looked at short pea plants and tall pea plants and pea plants kind of full seeds and others with shriveled up seeds, and he blended them. He crossbreeded them. And so what he did, he took just the example your book uses is the white and the red pea plants. And what he did, he breeded the white and the red. And note that the red, the alleles or the genes, expressions of this genes are capital A's. So that's the more dominant form, whereas the white color, lowercase a's, is a recessive form. When he breeded them, he found that every offspring had an allele that included the capital A or dominant one, and then the recessive one as well. But because the capital A is dominant, all the pea plants were red in color. And then in the second generation here, he took two of these red pea plants, bred them, and found that one of them was white. So even though this white color was lost or not expressed in the second generation, it appeared in the third generation. So this is how he knew that there was some code or some, well, he called them alleles, that were this white color, this recessive gene. And that's how genetics really got started. Now, when we look at genes and chromosomes, so these genes are inside the DNA double helix. And this double helix is really a long spiral molecule that resides in the chromosomes. In the chromosomes, horses have 64 chromosomes or 32 pairs. They occur in pairs. Humans, we have 23 pairs or 46 chromosomes. Fruit flies have 4 pairs or 8 chromosomes. So it varies depending on the species. But these chromosomes reside in the nucleus of the cell. And this is where the genetic information and really what's going on here, different segments of this DNA double helix are called genes. And the genes are the segments of that DNA where there's code or there's information that's passed on to make proteins. And those proteins cause a cell to behave in a certain way, to run certain applications in that cell. Looking at what the components, right, that these phosphate backbones are really connected by these base pairs, and those base pairs are the nucleotides. And you've probably heard of these: adenine, thymine, cytosine, guanine. But the thing to note, and what I'm going to ask you about, is that the pairs, adenine always pairs with thymine, cytosine always with guanine, right? And so when there's mitosis, and we'll mention that in a moment, when the cell splits and makes it a copy of itself, depending on what is left exposed, for example, if you have a combination of A, T, C, G, then what's going to form next to it, the other end of the DNA double helix, will be a T, A, G, C. And so that's those pairs that come up there. DNA double helix molecule showing some of the more complicated forms of it, but that's the DNA double helix there. And then this idea of mitosis, again, all DNA data can be replicated by separating the two strands of the DNA molecule, and that's what we call mitosis. And encoded information is used as a template for construction of an identical copy of that original DNA sequence. And so the DNA this way is passed on from generation to generation. But remember, mitosis is occurring inside a cell, and you're making two cells with complete pairs of chromosomes. The other one we'll talk about is meiosis. And meiosis is actually the formation of a sex cell. In this case, we have a cell. It begins to break in two, and it's going to produce either an egg or sperm with just one strand of that chromosome information. And then in this case, a sperm will fertilize an egg, and then now the egg will get DNA information from both its male parent and its female parent, right? So it's getting information from both parents. Now it's incorporated into the one fertilized egg, and that's basically the formation of a sex cell. But then that leads to passing that genetic information to the offspring. And then remember, mitosis is just taking a cell that's divided. We're going to split the chromosomes evenly, and we're going to make two cells, but again, with the full complement of chromosomes. Now let's go back to this previous slide here. So sequence of base pairs change over time. And so, in other words, one way where change occurs is this through mutations. And it's changing that encoded DNA formation or information. And so how do these come about? Most of them, at least from what I've been reading, seems to, these mutations seem to be from errors in that DNA replication. Maybe when it's replicating, an A is replaced by a G, and so that's going to change some of the information, especially if this happens in portions of the genes that provide a function, that make proteins. Another way mutation can happen is through a damage to the cell, either through some sort of radiation or exposure to chemicals. That may be more, well, depends on geologic time. Exposure to pathogens and maybe toxins may have been of a lower probability in the geologic past than it is today. Most mutations seem to be, your book says random, but it's really these errors in the DNA replication right in here. And then the mutated DNA will replicate again and again and again, and these are how the cell changes over time. And they could lead to some pretty amazing characteristics and changes. And then creatures descended from a single organism can acquire very different characteristics. Mutations are changes in the chromosome or genes and in that heritable information. Your book mentions there's chromosome mutations, which are log segments or large segments of the chromosome, or these point mutations that affect a single gene. They may be harmful, beneficial, or have no effect. I won't have a whole lot of time to talk about it in this class, but there are regions of the genes that are called introns and exons, and the exons are parts of the gene that actually do provide encoded information for producing proteins, and that causes the machinery of the cell to work and function in a certain way. But these introns are regions that really don't do anything. They don't code anything. They're just there maybe to pass a signal on at some point. But they do have some function, maybe in a position in the RNA sequence. They can also have mutations. And in fact, one of the ways we track speciation, at least trying to trace back common ancestors, is looking at the number of mutations that are occurring in those silent genes, a silent portion of the genes. Remember, these silent portions are not affected by natural selection. It's not going to affect the physical character of the organism. So that mutation in the gene will not have effect on the success or the fitness of the organism. But you can trace it. There's quite a bit of evidence now in research pointing that it's a pretty steady state rate of change. And we can actually subscribe numbers to it and figure out when species diverge from each other, especially closely related species like grizzly bears and polar bears. We can sort of figure out when they last shared a common ancestor based on the rate of these mutations in these silent intron gene portions of the gene. But anyways, that's a whole different lecture. Now, looking at this slide here, sexual reproduction and mutations account for most variations in populations. Your book mentions genetic drift, and it just says a random change, but it really doesn't go into any detail on it. This is a chance. This organism that may go on may not be the most fit to go on, but you can think of it as the most lucky. And the scenario here is we got a bunch of bugs here on the ground, some brown, dark ones here, some white ones. And let's say they're migrating across the cement, and you're riding your skateboard or your scooter across the sidewalk there, and you run right over and you smash a bunch of them. And let's say you smash all 6 of these. So that means these remaining 4, or 3 in this case, go on to have babies. Because remember, the key thing about evolution is being able to reproduce. And so they're passing their genes on here, and you'll see that the next generation, literally there are no dark colored bugs. So that's an example of this genetic drift where some random chance, something out of the control, nothing to do with fitness or natural selection, just some random scenario leads to the elimination of an allele. In this case, the allele that's being eliminated is the dominant allele, which is capital A in this case here, and only the recessive remains, and we have only the white or the light-colored bugs here. So again, it's a random change. It's due to chance. Affects small populations more than large populations, right? So the smaller the populations, the more enhanced this genetic drift will be. And it's not due to environmental challenges. Again, it's just a random occurrence. As opposed to natural selections, remember, this is due to some environmental challenge, something that the organism has to be adapted or have the right traits to be able to get through. It's directional generation of adaptive traits and adaptations. Again, the non-survival alleles fit and operates on the allele. And there's an increase in genetic variation. Now for species and speciations, so remember, species are natural populations of similar individuals that can interbreed and produce fertile offspring. And it's really about the gene flow, being able to pass your genes on to the next generation. And the only way to do that is by having viable offspring, because they're going to be able to pass your genes on to the subsequent generation and so on. This type of sexual reproduction doesn't really work with asexual organisms. So your book goes a little bit talks about bacteria a little bit, but we're just going to focus on this really enhanced form of evolution, which is the sexual reproduction here. Speciation is the formation of a new species from an ancestral species. Now, what can cause speciation? Here are two ideas. One is this geographic isolation. So it's also known as allopatric speciation. And species start because populations were prevented from interbreeding by some geographic isolation. So either there's a river, in this case a river is, here we have a population of beetles, and then the river separates them, and we see that the beetles on this north side of the river cannot breed with the beetles on the south side, and eventually they're isolated long enough where they have different characteristics where when they come back together they can't breed anymore. And so that's an example of this allopatric speciation. Or we can get maybe a reduction in gene flow. Remember, it's all about the gene flow. Here we have a huge species that's covering a huge area, like let's say the entire continent of North America. And it's very difficult for species on, let's say, the west side of the continent to breed with that same species on the east side. So they essentially, even though there may not be a barrier, they're going to be isolated from each other, and they're going to basically have zero chance of mating. And so they may develop into their own particular species, because selective pressures of that particular area where they live in on opposite ends of the domain would alter the gene frequencies in the groups. Again, you're changing that gene frequency. And so eventually the resulting population will be different. So that's a form of speciation. And so your book mentions this idea of microevolution. So these are small scale changes in how often that particular gene occurs. In this case, let's say we have brown beetles and green beetles. And when you first look at these beetles, you see that there's about 42% brown and about 58% green. And then over time, maybe 5 years go by and you remeasure these beetles and you find that 95% are actually green in this case here, and 5% are brown. So here we see a change. So that is a change in the frequency of the gene. In this case, you're seeing more green, 95 green, and only 5% brown, because these are mostly green here. So what are these mechanisms for microevolution? What's causing these small scale changes that eventually lead to speciation, to this macroevolution? So we could have mutations. Maybe more green genes occur because brown genes are mutating to become green genes. Or maybe there's a migration, an influx of green beetles into the population where they're passing on that green allele. Maybe there's a genetic drift, some random elimination of the brown gene. Or natural selection. Let's say a bird begins to eat all the brown beetles because these beetles are living in green grass and the brown beetles seem to stand out more.